this lecture on phospholipids and cholesterol. At this point, we've already looked at the fatty acids, and what you should be working on is chemically and structurally, what are they? Specifically, things like palmitate, linoleic, linolenic, the fatty acid structures in general, and those specific examples we covered in the previous lecture. We also talked about fatty acids and triacylglycerides and what purposes do they serve. So you want to take some time to review that. You want to know the synthesis of palmitic acid. You also want to understand beta oxidation of palmitic acid. Storage and mobilization of fatty acids we covered in detail, as well as regulation, at least hormonal regulation. We didn't cover obesity and type 2 diabetes, so you don't need to worry about that. For this lecture, and for the sections in your book, what you want to really be able to do are discuss phospholipids and what they do, and this includes structurally. The meat of this lecture is really cholesterol. How is it synthesized? How is that synthesis regulated? And then very briefly, we're going to talk about prostaglandins and thromboxanes and what physiological effects these have. So, in a brief overview, what is happening? We've already seen some of this. So, as we saw triacylglycerols will get mobilized from adipose tissue or from diet. You can get these from diet. And lipases will release free fatty acids that then travel to mitochondria and are broken down through beta oxidation to acetyl-CoA. On the other hand, the glycerol molecule itself can be converted to glycerol 3-phosphate, and we'll talk about that a little bit later on, and that's a part of glycolysis. So in humans, lipids are going to fall into two major categories, and those are nonpolar lipids, so these really are not going to be soluble in water, and that includes the triacylglycerols and the cholesterol esters, which we're going to talk about cholesterol more today. And polar lipids, which are amphipathic, and that means that they are capable of interacting with water and with hydrophobic solvents as well, and this includes the phospholipids. So what do these things do? And by these things, we're talking more about the phospholipids. Well, they make membranes. You've probably heard of the phospholipid bilayer. That's exactly what this is talking about, a membrane, where the core of the membrane is hydrophobic. That's where the phospholipid tails are. And we have a phosphohead group that's exposed to water. So they make membranes. They're also vital for cell-to-cell recognition. Phospholipids are often labeled with sugar molecules, that's glycosylation, and those serve as markers on the surface of cells. These markers can also communicate to cells to stop growing, so that when a population of cells comes into contact with each other, those cells know to stop growing because they recognize the signals, the glycosylation patterns carried by the phospholipids on the cell surface. Similarly, signals such as this can be used for phagocytosis, for phagocytes to consume and engulf debris or decaying, dead cells, that sort of thing. Phospholipids are vital for the conduction of nerve impulses, and they will be hormones. So things like prostaglandins and thromboxanes and other materials that might be derivatives of phospholipids and or cholesterol as well. So phospholipids follow the sort of same concept as the triacylglycerols. Okay, so we have two major classes of lipids here. We're talking about triacylglycerides, which we already covered previously, and the glycerophospholipids. Now, the glycerophospholipids come from a glycerol backbone, just like we saw with the triacylglycerides. In this case, two of these hydroxyl groups will be substituted with fatty acyls as opposed to all three. In a triacylglyceride, you'll remember all three of these hydroxyl groups are substituted with fatty acyl, and that's why the triacylglyceride is not soluble in water, or at least not terribly soluble in water. Phospholipids, on the other hand, are going to have two of these substituted by fatty acyls, and the third will be substituted with a phosphate group, and that phosphate group could be further substituted with other moieties such as sugars. That would be glycosylation. So glycerol, let's look at the glycerol for just a moment. It's a fundamental structural element of glycerophospholipids. Neutral triacylglycerols and charged ionic phospholipids have significantly different properties and functions. So here's a few examples of just some common phospholipids. I'm just showing you the generalized structures. So here we have our glycerol backbone. We see that's highlighted here in gray. And the fatty acyls are listed as R1 and R2. These are just going to be some sort of fatty acyl chain. And then we have our phosphate group here. You can see that phosphate, phosphate, and phosphate. And then we have some sort of labeling. So this is phosphatidylethanolamine. So here's the ethanol amine. Here's phosphatidylserine, where we have the amino acid serine associated with the phosphate group. Or this phosphatidylcholine or lecithin. We're going to get to lecithin a little bit more in a moment. So you can see a phospholipid can have some pretty distinct structures, mostly associated with substitutions on the phosphate, although the fatty acyls can be a little bit different as well. So membrane phospholipids, this is by far the highest concentration of phospholipids. They play structural and functional roles. A couple of examples are about half the mass of a red blood cell is membrane. And then there's an enzyme, beta-hydroxybutyrate dehydrogenase. This is on the inner mitochondrial membrane that requires the cofactor phosphatidylcholine. If you recall, here's the phosphatidylcholine as well. Nothing else will substitute. It's very, very specific for that phosphate. So we can have cofactors, structural roles, membranes. Phospholipids are found in a variety of places. But what I want to just zoom in on, because it's not intuitive, is the lung. Your lung function requires dipalmitoyl lecithin, which is a phospholipid, and this dipalmitoyl lecithin represents 80% of the phospholipid in the extracellular fluid of the lung. Now, this is a surfactant. If you recall, surfactants are, like a detergent, break up surface tension of water. So in your lung, you have a fine coating of fluid layer in your lung that, by having this surfactant, maintains the flexibility of the alveolus, and this prevents a problem called atelectasis. So here's an example of that. So we have the alveolus, and during expiration and inspiration, the alveolus inflates and deflates, inflates and deflates. Now, if you don't have the surfactant, then what can happen is the surface tension of the water can cause water to pull in on itself, which can draw the alveolus into collapse. So without the surfactant, you get atelectasis, and that's this, where the alveolus of your lung has collapsed and can no longer inflate properly. This causes a problem termed respiratory distress syndrome. This is found in neonatal, so this is a neonatal problem, and accounts for 15 to 20% of neonatal deaths in Western countries. This is somewhat less in developing countries. Only strikes premature babies. An instance varies directly with degree of prematurity. So a premature baby is going to have greater chances of respiratory stress syndrome depending upon how premature the infant is. And the cause of it is due to deficiency of the pulmonary surfactant, and this can be measured by the lecithin-sphingomyelin ratio in amniotic fluid. It's very diagnostic. 2.0 is characteristic of term birth, so 9 months. However, notice that even a 1.9 ratio, shown here, 1.9, will give you a 40% chance of the infant having this syndrome. 1.5 to 1.9 is 40%, less than 1.5, 75% chance the infant will have the syndrome. And the problem is the type 2 cells of the lung have not fully matured, and it's these cells that produce the surfactant. Therapy in this case is simply replacement surfactant until the type 2 cells mature. It resolves over time. It can occur in adults due to death of type 2 cells, and this can happen from chemotherapeutics. So many cancer treatments are going to cause this type of issue, as well as immunosuppressants can have this side effect. So here we go, ladies and gentlemen. Here it is, cholesterol. you should be able to draw this structure. You should know this ring system very, very specifically. Cholesterol, or 5-cholestyl 3-beta-ol, has a very signature structure. This three six-membered rings joined together with this five-membered ring is so easy to spot. Now you may see sometimes people say, well, this hydroxyl group allows cholesterol to be soluble. No, cholesterol is ridiculously insoluble. It does not go into water. You may also occasionally hear the media say there's such things as good and bad cholesterol. There is not. There is only cholesterol. What they're talking about when they say good and bad cholesterol is really low density lipoprotein and high density lipoprotein, and that's something else entirely. There is only one molecule called cholesterol, and this is it. I like to bring up this quote just to show how important cholesterol is. Cholesterol is the most highly decorated small molecule in the history of biology. 13 Nobel Prizes have been awarded to scientists who devoted major parts of their careers to cholesterol. Ever since it was isolated from gallstones in 1784, cholesterol has exerted an almost hypnotic fascination for scientists from the most diverse areas of science and medicine. Cholesterol is Janus-faced molecule. That means it's two-faced. The very property that makes it useful in cell membranes, namely its absolute insolubility in water, also makes it lethal. And this was from Michael Brown and Joseph Goldstein on the occasion of their receipt of the Nobel Prize for elucidating the control of blood levels of cholesterol. So just how evil is cholesterol? Well, cholesterol is actually a critical material. You have an absolute requirement for cholesterol. It is important in cell membranes, bile salts, and hormones. As an aside, it is entirely unlikely that cholesterol will kick puppies, embezzle funds, or participate in telemarketing programs. So, cholesterol is used in bile salts and hormones. Bile salts derive from cholesterol, and bile salts are used to dissolve fats. So guess what? Bile salts can dissolve cell membranes. So guess what? Your gallbladder makes bile salts. Your gallbladder is, of course, comprised of cell membranes. Therefore, bile salts can dissolve the gallbladder. Therefore, you also need cholesterol to protect the gallbladder from the bile salts it makes. So bile phospholipid solubilized cholesterol. We see these made in the liver, and we can have problems in the gallbladder as a result of this. Same problems I was just talking about. So bear in mind, you need cholesterol for cell membranes. Of course, too much cholesterol in cell membranes can make them too rigid. You need cholesterol for hormones. You need cholesterol for bile salts to dissolve fats. It's a very important and ubiquitous molecule. So where do we get cholesterol? Well, you get it from diet, number one. Uh, and importantly, de novo synthesis. This means just making it. Virtually all of the cells of the body can make cholesterol. They all can practice a synthetic pathway. It is absolutely abundant in myelinated structures such as the brain and nervous system. And importantly, we have no capacity to oxidize cholesterol down to CO2 and water in humans, unlike sugars and even fats, which we can kind of juggle around. We can't do that with cholesterol. We can't break it down to carbon dioxide and water. It just doesn't work. So how do you get rid of it? Excretion is through the liver and gallbladder as bile acids into the intestine, where it's eliminated through the fecal route. A few more uses of it. Well, we've already talked about the bile acids. So you need this for absorption of dietary triacylglycerols and fat-soluble vitamins. It's a precursor to steroid hormones, progesterone, corticosteroids, aldosterone, estrogen, testosterone, and you need it for the formation of vitamin D. So with that, let's start talking about the synthesis of cholesterol. It requires acetyl-CoA. Fortunately, we know where to get acetyl-CoA now, at least a couple of ways to do it. There are a couple of others that we'll cover later. Three major stages. First, we have to make the building block, which is isopentenyl pyrophosphate. That's our key building block. We've seen this idea of building blocks before with malonyl-CoA. Then we're going to move to the lumen of the ER, where we're going to condense 6 molecules of isopentenyl pyrophosphate to form squalene. And then also in the lumen of the ER, stage 3 is cyclization of squalene and conversion to cholesterol. I'm going to kind of do this a little bit backwards. I'm going to get you to the building block first, but then I'm going to skip to the end and work our way backwards, which makes it a little bit easier to see how cholesterol is formed and how everything comes together. So we're going to start with acetoacetyl-CoA, and we're going to transfer the acetyl-CoA, the acetyl group from acetyl-CoA onto acetoacetyl-CoA. The energy for this transformation comes from the thioester bond of the CoA. This is going to be driven by the HMG-CoA synthase reaction, and our product is HMG-CoA plus a CoA molecule. So essentially we're just simply transferring acetyl group onto acetyl-acetyl-CoA. We get HMG-CoA. This requires a little bit of water as well, as you can see. And we get HMG-CoA and free CoA, coenzyme A. Now our next step, this is the actual rate limiting step of the process. We have HMG-CoA. We're going to use 2 NADPH, so this is obviously a reduction. From this we get mevalonate, free CoA, and NADP. This is HMG-CoA reductase. This is the rate limiting step. So we're not to the building block yet. We're at mevalonate, where we've converted HMG-CoA to mevalonate. Next. Okay, so from mevalonate to 3-isopentenyl pyrophosphate, we're going to do a series of phosphorylations. So we're going to take mevalonate and we're going to phosphorylate that. The enzyme that phosphorylates mevalonate is mevalonate kinase. That makes sense. Then we're going to phosphorylate the phosphorylated mevalonate. So we take phosphomevalonate and we phosphorylate that. The enzyme is phosphomevalonate kinase. Now you get 5-pyrophosphomevalonate. We have 5-pyrophosphomevalonate. We decarboxylate that, so that's going to release some CO2. And we get pyrophosphomevalonate decarboxylase as our enzyme. Notice very usefully the enzyme names tell you what they do. And by decarboxylation we get 3-isopentenyl pyrophosphate. That is our building block. So just reviewing the reaction to build the building block. We have mevalonate. We're going to spend an ATP using mevalonate kinase to make 5-phosphomevalonate. 5-phosphomevalonate using phosphomevalonate kinase gives you 5-pyrophosphomevalonate. Then we're going to use pyrophosphomevalonate decarboxylase. Notice we do add another, we do have yet another phosphorylation here, and this is the inorganic phosphate that's going to come off along with the CO2. That gives us this reaction. Here we see this electron pair hops down, the CO2 leaves, we get a double bond with this CH2 and a CH3. We have 3-isopentenyl pyrophosphate. This is our key building block. Now I'm going to jump ahead. Don't panic, we're going to jump ahead a little bit here. So you have isopentenyl pyrophosphate. Just set that aside for a moment, because we're going to work backwards. So our target molecule is squalene. Squalene is the key precursor of cholesterol. Let me just show you. So if we take squalene, which is a 30 carbon chain, there it is. You can see this is really, really close to the cholesterol structure. In fact, you see we have our ring system not quite there, but we can see it. So let's start at the end and build our way up. Farnesyl combines with farnesyl pyrophosphate combines with farnesyl pyrophosphate to make squalene. We lose 2 pyrophosphates in the process. It does require some reduction, and so we get an NADP and free proton. The enzyme that makes this is squalene synthase. So we take 2 farnesyl to make squalene. Remember, these are farnesyl pyrophosphate. This happens in the ER. Next, and we're starting at the bottom of this figure, we have farnesyl pyrophosphate. How do we get the farnesyl? Okay, so remember we need 2-farnesyl to make squalene. Okay, so we need to make farnesyl twice. How do we make farnesyl? Farnesyl comes from a molecule called geranyl, geranyl pyrophosphate. It combines with isopentenyl pyrophosphate, so there's our building block. We lose pyrophosphate off the geranyl, and we have now made farnesyl. This is done by geranyl transferase. That is the enzyme that makes it. Geranyl transferase makes farnesyl pyrophosphate. But how do we make geranyl? Okay, geranyl comes from a molecule called dimethylallyl pyrophosphate, and it combines with isopentenyl pyrophosphate. We lose the pyrophosphate off the dimethylallyl. Notice we keep the pyrophosphate on the isopentenyl each time. Dimethylallyl combines with isopentenyl pyrophosphate. The dimethylallyl loses the pyrophosphate. There's the energy for the reaction, and that creates geranyl pyrophosphate. So now we can go with dimethylallyl combines with isopentenyl to make geranyl. Geranyl combines with isopentenyl to make farnesyl. Farnesyl combines with farnesyl to make squalene. The enzyme that makes the geranyl is geranyl synthase. So now the question is, okay, where did the dimethylallyl pyrophosphate come from? Okay, this is the very beginning. We take an isopentenyl, so there's one isopentenyl, and we isomerize that isopentenyl pyrophosphate, we isomerize that to dimethylallyl pyrophosphate. The enzyme is isopentenyl pyrophosphate isomerase. So dimethylallyl, 3,3-dimethylallyl pyrophosphate is an isomer of isopentenyl pyrophosphate. So there's one isopentenyl. We then take a second isopentenyl and combine it with dimethylallyl. So we now have spent 2 isopentenyl. That gives us geranyl. Geranyl combines with an isopentenyl pyrophosphate, there's 3, to make farnesyl. Do that twice. That's 6 isopentenyl pyrophosphate spent to make 2 farnesyl pyrophosphate. The farnesyl pyrophosphates combine, burning both pyrophosphates, to give you squalene. And squalene is our precursor to cholesterol. So there it is. By going backwards, hopefully you see it a little bit more clearly. So we get HMG-CoA reductase makes mevalonate, mevalonate kinase makes phosphomevalonate, phosphomevalonate kinase makes 5-pyrophosphomevalonate kinase, pyrophosphomevalonate decarboxylase removes the decarboxylate, so removes the carboxylate from mevalonate to give you 3-isopentenyl pyrophosphate. That's our building block. Isopentenyl pyrophosphate is isomerized to 3,3-dimethylallyl pyrophosphate. Dimethylallyl pyrophosphate is combined with isopentenyl pyrophosphate to give geranyl pyrophosphate. Geranyl synthase does this. Geranyl pyrophosphate combines with another isopentenyl pyrophosphate, so we've spent 3 using the enzyme geranyl transferase to create farnesyl pyrophosphate, 2-farnesyl using squalene synthase, and this is the same showing the same thing. Farnesyl pyrophosphate plus farnesyl pyrophosphate lose both pyrophosphate, do a little bit of reduction, squalene synthase, you get squalene. Okay, so there's squalene, 30 carbons. Now, from squalene, we have to go from squalene to squalene epoxide. Notice this is a reduction and dehydration as well, and that gives us our cyclization to prosteryl cation. Prosteryl cation then goes to lanosterol, then 19 steps later, we go from lanosterol to cholesterol. So this is a ridiculously long synthesis, and you don't need to know it. If you can get to squalene, you've done well. Okay, squalene is our target molecule. From there, we assume cyclization to cholesterol. And that's it. That's it for cholesterol synthesis. So, how do we regulate this? Cells regulate cholesterol synthesis at the rate limiting step, which is HMG-CoA reductase. Remember, that's the enzyme that makes mevalonate. So how do we control HMG-CoA reductase? Well, it's a classic non-competitive system. Cholesterol itself, the product of the pathway, is an inhibitor of HMG-CoA reductase. So we see that non-competitive inhibition. We know we don't influence binding of the HMG-CoA to the enzyme, but we do turn off catalysis. So we would expect no change in Km, and we would expect a loss of Vmax upon creation of cholesterol. So that's one mechanism. It turns out HMG-CoA reductase is very highly regulated. So first we have feedback regulation. We also have genetic regulation for the mRNA. What this means is the sterol regulatory element binding protein, you don't need to know that, actually controls the rate of transcription. We also control, just in case that wasn't enough, rate of translation. So that's inhibited by mevalonate-derived intermediates and dietary cholesterol. So we're controlling the rate of creation of mRNA. Then dietary cholesterol and presence of mevalonate intermediates also controls the rate of translation. So we're not even making the protein. So first we're not making the message, then we're not making the protein. With cholesterol, we're also not activating the protein. We can also control the rate of degradation of the enzyme itself. So the membrane domain alters conformation in response to sterols. And that conformational change enables proteolysis of the enzyme. So the enzyme can just be degraded. And in case all of that was not enough, there's phosphorylation. So we can have a protein kinase that is activated by AMP, and it will just phosphorylate the enzyme HMG-CoA reductase and turn it off. All of this together is a greater than 200-fold change in the activity of the enzyme. This is an astoundingly regulated enzyme. So just for fun, let's take a look at vitamin D. Interestingly enough, a lot of people don't realize human beings actually do photosynthesize. There's one thing we can photosynthesize, and that's vitamin D. This is a very slow reaction. It requires ultraviolet light, and we get conversion to vitamin D is not enzymatically driven. It's very, very slow. And all you need to maintain proper levels of vitamin D in your body are 10 minutes of sunlight exposure per day on your hands and face. That's it. That's all you need. So how does it work? Well, here's our cholesterol. And a simple strike with ultraviolet light opens up the ring, so we see electrons transfer over and creating this double bond. That causes the electrons from this double bond to move to here. We lose our aromaticity, which causes these electrons to hop over to form a double bond here. The ring is now open. That's pre-vitamin D3. Now, through a non-enzymatically driven process, we get cholecalciferol, and then you get, that is vitamin D3, and then over time, that can become calcitriol. Which is just a derivative we use elsewhere. But here is our vitamin D3. So our reaction is an ultraviolet light driven reaction from cholesterol, or 7-dehydrocholesterol, which results in a ring opening, which results in pre-vitamin D3 converting to vitamin D3. Lastly, and very briefly, let's look at the prostaglandins. These also derive from cholesterol. They won't even be recognizable as cholesterol anymore. And what do? They promote contraction of intestinal and uterine muscle. They lower blood pressure. They're highly important medically and produced by almost all cells. But notably, they're not produced by red blood cells. These are not stored. They're synthesized, used, and removed. Control of these things is not very well understood. So your basic prostaglandin, you can see, is a huge, basically fatty acid. This is prostanoic acid. We have our 5-membered ring. That's about all that you see remaining of the cholesterol. It does have chirality. It's 20 carbons long, and that's it. It's a very simple, very small molecule. But what does it do? Well, They are mediators of tissue inflammation in response to tissue damage. And inflammation is really a terrible thing your body does. But they cause the prostaglandins cause vasodilation. That's the redness and heat, swelling and edema due to capillary leakage. This is treated with corticosteroids to inhibit prostaglandin production. They also cause T cell migration, so these are useful in wound healing, and they enhance pain caused by histamine and bradykinin. So they're really just kind of stirring up everything here. They're generated in response to fever. But interestingly enough, synthetic forms are useful for patients with peptic ulcers, as it inhibits gastric acid secretion. So while they do some things that are of questionable value, such as inflammation, they do promote T cell migration. And pain is a questionable thing, whether or not it's good to have pain because you know to stop doing whatever it is you're doing. On the other hand, pain is not something we really want to experience. And what prostaglandins do and how they work is poorly understood. However, they are very active in a wide variety of physiological responses. And with that, that's it. So be sure to go back and study, especially the cholesterol synthesis part, be able to work through it, understand those chemical reactions and the enzymes involved. As far as you may be asking, well, what about cholesterol degradation? Simple, we don't really do it. There's conversion of cholesterol to other materials, such as the bile acids that are eliminated, but we don't really catabolize cholesterol down the same way we do sugars and fats, or even proteins. So you don't even have to worry about that. But with that, go forward, study, and be prepared for your workday challenges.